students, in this class I am going to explain about DNA to protein. That is how the protein is synthesized on the basis of the information stored in the DNA. Since on the last turn I had told you that DNA is the storehouse of the genetic information. So in the translation process, the protein synthesis takes place. The process in which the genetic information contained in DNA is transferred to mRNA that programs the ribosomes for the synthesis of protein molecule can be called as translation. This already I have explained in the central dogma, that the genetic information which is contained in the DNA is transferred to mRNA, and this mRNA programs the ribosomes for the synthesis of protein molecule. Now in this process, the particular nucleotide sequence which is present on mRNA is translated into a particular amino acid sequence, forming a polypeptide chain. That is the nucleotide sequence. You know what is the nucleotide sequence? It will be whatever it will be present on the mRNA, it will be translated into a particular amino acid sequence, and thus this will form a polypeptide chain. Thus you can say that DNA controls all the life processes by directing and controlling the synthesis of various kinds of proteins. This question comes that how the DNA controls all the life processes. It controls it by directing and controlling the synthesis of various kinds of proteins, because the information is stored in the DNA only. Hence, it is through mRNA the genetic information is passed on to protein with the help of a coded message given by DNA. The DNA provides the coded message, and through mRNA the genetic information is passed on to protein. This process translation, I hope you understood, that on the basis of the information which is stored in the DNA, it is transferred to mRNA that programs the ribosome for the synthesis of protein molecule. Now the next is the genetic code. The relationship between the nucleotide bases and the amino acid. Whatever the nucleotide bases will be there, accordingly the amino acid will be synthesized. For this, children, please refer figure 24. It is given in your book about the information regarding the amino acid. Now here you can say the order of base pairs along the DNA molecule controls the kind and order of amino acid. Whatever the base pairing will be there in the DNA molecule, accordingly there will be the order of amino acid and its kind, which is found in the protein. That is, the order of base pair along the DNA molecule controls the kind and order of amino acid found in the protein. This specific order of base pair is called genetic code. This specific order is called as genetic code. In one line, it is the specific order of base pair, it is called genetic code, and this makes an individual unique because this base pair order differs from individual to individual. Thus, the genetic code is a sequence of nitrogenous bases along a sugar phosphate strand of DNA molecule. What is genetic code? It is a sequence of nitrogenous bases along a sugar phosphate strand of DNA molecule. This was given by Marshall, Nierenberg, Severo, and Hargobind Singh Khurana. Now here codon. Codon is a sequence of 3 nucleotide bases which is a triplet that codes for a particular amino acid. It is a sequence of 3 nucleotide bases that code for a particular amino acid. For example, if mRNA has sequence of triplet code UUU, then the corresponding amino acid in the protein would be phenylalanine. Children, please refer that table. I will be sending that table also. So whatever will be the codon, it will be in the form of nucleotide bases, which will be triplet, and this will code for particular amino acid. Then nature of genetic code. First is genetic code is triplet. For example, U, and it will code for phenylalanine. Genetic code is universal. All the known living systems use the nucleic acid and the same 3 base codons to direct the synthesis of protein. So that is why it is same for all. It is universal. That is, all known living systems use the nucleic acid and the same 3 base codons to direct the synthesis of protein from amino acids. Next is genetic code is non-overlapping. That is, same letter is not used for 2 different codon. Same letters is not used for 2 different codons. It is commaless. That is, message is read directly from one end to another. No punctuation is needed in between. Next is the genetic code is degenerate. Means more than one triplet sequence could code for a specific amino acid. More than one triplet sequence. In the table you will be coming across with this situation. Could code for a specific amino acid. Example, GUU, GUC, GUA and GUG. They all will code for valine. So that is why genetic code is degenerate. More than one triplet sequence could code for a specific amino acid. Genetic code is non-ambiguous, that it is very specific. That is, sequence code for only one amino acid. That is given over that particular sequence will code for only one amino acid. It is given in the table. Now there are 2 types of codons, namely chain initiation codons like AUG, methionine, GUG, valine. These are the chain initiation codons. Whereas chain termination codons are UAA, UAG, that is the tyrosine, UGA, that is the tryptophan. So these are also known as nonsense codons, since they do not code for any of the 20 essential amino acids, but they have their merit also because they help in the release of polypeptide chain from the ribosome. So children, today you have studied about the DNA to protein, that is the translation process in which the protein synthesis takes place according to the information contained in the DNA. Then second, you have read what is the genetic code. Genetic code, it is a sequence of nitrogenous bases along a sugar phosphate strand of DNA molecule. This is 1 mark definition. Now in the contribution of the scientists, this Marshall, Nierenberg, Hargobind Khorana, their name is coming. So you remember that they discovered the genetic code. Then after that, codon, sequence of 3 nucleotide bases. 3 nucleotide bases that code for a particular amino acid is called as codon. For example, UUU will code for phenylalanine. The nature of genetic code, it is triplet, as you can see over here. It is universal. Then after that, it is non-overlapping. Same letter is not used for 2 different codons. Accordingly, same letter is not used for 2 different codons. It is commaless, that is, message is directly read from one end to the another. There is no punctuation needed. Here you can say that example, no punctuations needed between any 2 codons, and it means that after one amino acid is coded, the second one will be automatically coded by the next 3 nitrogenous bases till the stop codon, which marks the termination of polypeptide chain, is coming. So that is why it is commaless. In the protein synthesis, it will be more clear to you. Then after that, genetic code is degenerate, means more than one triplet sequence could not code for a specific amino acid. They all triplet sequence will code for valine only. Then after that, the genetic code is non-ambiguous. that is, it is specific. And the last one, chain initiation codon, remember their name, AUG and GUG, methionine and valine. And chain termination, UAA, UAG, UGA. UAA, UAG is the tyrosine and UGA is the tryptophan. They are called as nonsense codon because they do not code for any of the 20 essential amino acid, and they help in the release of polypeptide chain for ribosome. Children, this molecular basis of inheritance I am taking each and every day stepwise, because this is the concept to be remembered stepwise, because otherwise you will not be able to understand the second step. So that is why I am taking that only what you can understand within a period, within a single period. So even if you have any problem, you are always requested to ask your problem on my number. And side by side, this one evolution concepts, they are very easy, so you go through with that. And there also, if something you are lacking, please ask. Thank you.